Get this, scientists are using AI to study viruses, but not in the way you might think.
Oh,
Instead of, focusing on when the AI gets things right, they're kind of looking at its mistakes.
interesting.
Yeah. It's called Genome Misclassification Network Analysis, or GMNA for short.
Okay.
And we're diving deep into a research paper that applied this method to, like, over 500,000 SARS-CoV-2 genomes.
Wow.
Turns out these AI flubs can reveal some pretty surprising secrets about how viruses evolve and spread.
It's a bit like turning lemons into lemonade, right?
Yeah, exactly.
Those misclassifications actually provide clues about the relationships between different viral genomes.
Okay, so before we, get to the AI and all that, let's just
Ah.
make sure we're all on the same page.
Sure.
What exactly is comparative genomics? I know it, involves comparing genomes, but paint me a picture.
So imagine you're trying to figure out how closely related different species of birds are.
Okay.
You might compare their beak shapes or wingspans or
Yeah.
even the songs they sing. Comparative genomics is kind of similar, but instead of physical traits, we're comparing the genetic code, the DNA of different organisms.
So we're looking for those family resemblances, but in the DNA code.
Exactly.
But how do scientists actually compare those genomes? Is it like lining them up side by side and circling the differences with a red pen?
That's a pretty good analogy.
Yeah.
Traditional methods often involve sequence alignment,
Yeah.
which is kind of like laying two genetic sequences next to each other and marking where they match up.
I see.
Imagine comparing two long strings of beads,
Yeah.
looking for similar patterns.
So-
There are well-established tools for this, like
Gotcha.
BLAST and Clustall.
Okay, so with millions, even billions of letters in those DNA sequences, that sounds like a pretty tedious task.
It can be, especially with the massive data sets we have today.
Right.
And those traditional methods sometimes miss more subtle relationships, It's like trying to understand a complex family saga just by looking at a basic family tree. You need more information to capture all the nuances.
Okay, so how do scientists get around those limitations?
Well, they've developed alignment-free methods
Oh.
that use different approaches, like analyzing conjugate statistics.
Ganger's statistics.
Think of conjures as short words within those long DNA sequences.
Okay.
By comparing the frequency of these words across different genomes, we can get a sense of their relatedness.
Okay, that makes sense.
Yeah.
But I'm guessing there are still, challenges.
Sure.
Enter deep learning, right?
Yeah. It's
Everyone's talking about deep learning these days.
all the rage.
Give me the rundown. How is it being used in genomics?
Deep learning is like having a super-powered detective on the case,
Okay.
It uses complex algorithms inspired by the human brain to sift through enormous amounts of data and find patterns that we might miss otherwise.
Interesting.
In genomics, deep learning models can identify specific DNA sequences that control gene activity or even predict how genes will be expressed in different contexts.
Wow. So it's giving us a deeper understanding of how those genes actually function.
Exactly.
But I'm guessing there are still, challenges even with deep learning.
Of course, deep learning models can be computationally expensive to train.
Right.
They need massive amounts of data and processing power.
I see.
And sometimes they struggle with extremely long genomes, like those of SARS-CoV-2.
Okay, so we've got these traditional methods in deep learning, both with their own, strengths and weaknesses.
Yeah.
That's where GMNA comes in, right?
Right.
What's the big idea behind this new approach?
GMNA flips the script.
How so?
Instead of celebrating when the AI classifies genomes correctly,
Right.
we're focusing on when it gets things wrong.
Wait, so we're intentionally looking at the AI's mistakes?
Yeah.
That seems a bit counterintuitive.
I know, right?
Why would we do that?
Because those mistakes can be incredibly revealing. The basic principle is this. If the AI struggles to distinguish between two genomes, it means they likely share some crucial features that make them look very similar.
So it's like those spot the difference puzzles.
Exactly.
If you can't find the differences, it means those two images are very closely related.
Yeah. And by analyzing those misclassifications,
Right.
we can build a network, a map of sorts, that reveals hidden relationships between different genomes.
That's pretty clever.
It is.
But how does this actually work in practice? Can you walk me through it?
Think of it like this.
Okay.
You have a group of people,
Okay.
And you're trying to classify them based on their hometown.
Right.
Now, if you have a really good AI,
Right.
it might accurately predict everyone's hometown based on their appearance or
Right.
accent or even their favorite foods.
Mhm.
But let's say that AI keeps misclassifying people from two specific towns.
Okay.
Let's say Boston and New York City.
Boston, New York. Okay.
It keeps mixing them up because culturally, those cities are quite similar. They're both bustling metropolises on the East Coast.
Right.
So the AI's mistakes are actually telling us something about the similarities between those cities.
Yeah, so that AI's mistakes are actually telling us something about the similarities between those cities.
And in GMNA, we take those myth classifications and use them to build a network.
Okay.
In this case, Boston and New York City would be strongly connected in the network because the AI frequently confuses them.
So we're essentially turning those, oops, I got that wrong moments, into valuable data points.
Exactly.
I'm starting to see how powerful this could be.
And the beauty of GMNA is that it's very flexible.
How so?
It can be used with different types of AI models,
Oh, okay.
depending on the complexity of the data.
Mhm.
It's like having a toolbox full of different AI-powered magnifying glasses,
I like that analogy.
each suited to a particular task.
Okay, so we've got the basic idea.
Right.
But let's get specific.
Sure.
You mentioned that this research applied GMNA to a massive data set of SARS-CoV-2 genomes.
They did.
Tell me more about that.
Right.
What were they trying to find out?
They started with over 550,000 SARS-CoV-2 genomes,
Wow.
collected from 198 regions worldwide.
That's a lot of data.
It is. Their goal was to understand how the virus was evolving and spreading across the globe.
And they used GMNA to do this.
Exactly.
Okay.
Remember those challenges we talked about with traditional methods and deep learning?
Right, right.
Well, they had to address those head on.
Like the computational complexity of dealing with those long viral genomes.
Oh, exactly.
Yeah.
They used some clever pre-processing steps to prepare the data for the AI models.
Like what?
Well, one technique they use is called commarization,
Commoditization, right?
where they break down the genome into those short words we talked about earlier.
Ah, so they're making it easier for the AI to digest those long genetic sequences?
Precisely.
Hmm.
They also use a technique called one-hot encoding,
Okay.
which is a way of representing those commers numerically so the AI can understand them.
Okay, so they prepped the data, they've got their AI models ready to go. What did they find?
Yeah.
What secrets did those AI misclassifications reveal?
One of the most striking findings was that SARS-CoV-2 genomes clustered very strongly by geographic region. The AI frequently misclassified genomes from neighboring regions, suggesting that viruses within those areas shared a lot of genetic similarities.
So it's like those viruses within a region are more closely related, like members of the same family.
Exactly, and the AI was having trouble telling them apart.
Were there other examples of this regional clustering?
Absolutely. They saw similar patterns in other parts of the world.
Mhm.
For instance, the AI frequently mixed up genomes from Canada, the US, and Mexico.
Interesting.
It's like the AI was saying, hey, these viruses are all hanging out in the same neighborhood. They probably have a lot in common.
So the AI is essentially mapping out these viral family reunions,
Yeah.
showing us which viruses are most closely related.
Yep.
But why would that be the case? Why would viruses from a particular region be more similar to each other?
That's where the connection to travel comes into play.
Okay.
And this is where it gets really fascinating.
Okay. All ears.
The researchers found a link between the AI's misclassification patterns and international travel data.
Wait, you're telling me that airplane travel is somehow leaving its mark on the genomes of viruses?
It is.
How is that even possible?
Well, think about it. When people travel, especially internationally,
Right.
they can carry viruses with them.
Okay.
This movement of people and the viruses they carry leads to a mixing of different viral variants across different regions of the world.
So it's like a global viral mixer fueled by international travel.
Precisely. And the more travel there is between two regions, the more their viruses get mixed up, making them harder to classify based on their region of origin.
So the AI is basically telling us, through its mistakes, that travel is playing a role in shaping the genetic diversity of viruses like SARS-CoV-2.
Exactly, and this finding has some important implications for understanding how viruses evolve,
Right.
how new variants emerge, and how we might prevent future outbreaks.
This is all blowing my mind.
It is fascinating.
So we've got these AI misclassifications revealing hidden connections between genomes and showing us how travel impacts viral evolution.
It is.
But there's one thing I'm still wondering about.
What's that?
You mentioned earlier that a perfect AI wouldn't actually be helpful for this kind of analysis.
Right.
Why is that? Wouldn't we want the AI to be as accurate as possible?
That's a great question, and it gets to the heart of why GMNA is so innovative.
Okay.
If the AI were perfect,
Right.
always classifying genomes correctly,
Yeah.
we wouldn't have any misclassifications to analyze.
I see.
No network, no map, no insights into those hidden relationships.
So some misclassifications are actually good.
Exactly.
It's like that old saying, we learn from our mistakes.
Right.
Those mistakes, those misclassifications are the key to unlocking the secrets hidden within the data.
That's a good way to put it.
Okay, so there's a sweet spot where the AI is good enough to be reliable but makes enough mistakes to give us valuable data points.
Right.
But how do researchers find that sweet spot?
Well, that's where things get even more interesting.
Okay, lay it on me.
The researchers in this study came up with a clever approach called the Leave One Class Out model, or LOCO for short.
LOCO.
LOCO.
It sounds like we're going a little crazy here, but I'm definitely intrigued.
It is a bit out there.
What is this LOCO model all about?
Imagine you're training the AI to classify those viral genomes based on their region of origin.
Okay.
In the LOCO model, you intentionally leave out all the genomes from one specific region during the training process.
So it's like the AI has a blind spot for that region.
Exactly.
Okay.
When you then test the AI using genomes from that excluded region,
Right.
it's forced to misclassify them because it's never seen genomes from that region before.
That's sneaky, but brilliant.
It is a clever approach.
So they're essentially guaranteeing a certain number of misclassifications, which become the raw material for building that network.
Precisely.
Okay.
And this method helps to address that trade-off we were talking about.
Right.
By intentionally creating misclassifications.
Right.
They can ensure they have enough data points to build a meaningful network while still maintaining a reasonable level of overall accuracy in the AI's performance.
Okay, so we've covered a lot of ground already. Comparative genomics, deep learning, and this fascinating GMNA method that uses AI mistakes to uncover hidden connections.
We have.
But I'm eager to hear more about what they found regarding travel and viral evolution.
Okay.
What specific insights did they gain?
Well, for that, we'll need to dive even deeper.
Let's do it.
Huh?
All right, so we've established that AI can, make some pretty insightful mistakes.
Yeah.
But how do the researchers actually connect those misclassifications to, like, real-world travel patterns? It's not like they asked the virus, hey, where have you been hanging out lately?
Right. They needed a more concrete way to measure travel's impact on viral spread.
Mm.
And they found it in the official airline guide,
The what?
the OAG.
OAG.
Think of it like a giant database tracking flight routes and passenger volumes all over the world.
So they use this airline data to track the movement of people,
Yeah,
which we can assume reflects, at least to some extent, the movement of viruses as well.
exactly.
Makes sense.
They took this OAG data and built a network representing international flight patterns.
Okay.
Picture a web of connections spanning the globe, with each country represented by a node,
Right.
and the thickness of the lines between them representing the volume of air travel.
Okay, so we have a network of viral genomes based on those AI misclassifications.
Right.
And now we have a separate network showing global flight patterns.
Yeah.
What did the researchers do with these two networks?
They combined them, of course.
Oh, okay.
They looked for correlations between how well connected a country was in the flight network, its centrality, and how often its viral genomes were misclassified with those from other countries, its aggregated indistinguishability score.
Hold on. Let's unpack those terms a bit.
Sure.
Centrality in the flight network. Does that basically mean how much of a travel hub a country is?
Exactly. Imagine a social media influencer with millions of followers.
Okay.
They have high centrality in the social media network because they're connected to so many people.
I see.
Similarly, a country with a lot of international flights coming and going,
Right.
like the UK or Dubai,
Yeah.
would have high centrality in the flight network.
Got it. So the more connected a country is in terms of travel, the higher its centrality.
Right.
Now, what about aggregated indistinguishability? That sounds a bit complex. Think of it like a group of friends who are always getting mistaken for each other.
Okay.
Maybe they have similar hairstyles or fashion sense or
Right.
mannerisms.
Mm-hmm.
The more often people mix them up, the higher their indistinguishability score.
In this case, the aggregated indistinguishability score for a country reflects how often its viral genomes were confused with those from other countries.
Okay, so a country with a high aggregated indistinguishability score has viruses that are often mistaken for viruses from other places.
Yeah.
It's like they've got a less distinct look because of all that mixing.
You got it. Now, here's where it gets really interesting.
Okay.
The researchers found a strong correlation between these two factors. Countries that were major travel hubs, those with high centrality in the flight network,
Right.
tended to have higher aggregated indistinguishability scores in the viral genome network.
So the more connected a country is in terms of travel, the more its viruses resemble those from other parts of the world.
Right, right.
That makes sense, but why is this finding important?
Well, think about the implications for tracking and managing infectious diseases. It means that travel patterns are actively shaping the genetic landscape of viruses like SARS-CoV-2.
Right.
This knowledge can help inform strategies for disease surveillance, travel restrictions, and even vaccine development.
It really highlights the interconnectedness of our world, doesn't it?
It does.
It's a reminder that a virus outbreak in one part of the globe can quickly become a global concern.
Absolutely. These findings can help us predict how viruses might evolve and spread based on those travel patterns.
Right.
It's like having a glimpse into the future of viral outbreaks.
This is all fascinating, but let's go back to those variants of concern for a moment.
Okay.
Remember, these are the variants that tend to spread more easily or cause more severe disease or, evade our immune defenses.
Right.
The researchers found a link between these variants and those periods of increased genome indistinguishability.
They did.
Can you explain that connection a bit more?
Of course, remember that variants arise from mutations in the viral genome.
Right.
When you have a lot of travel and mixing of different viral lineages,
Yeah.
it creates more opportunities for those lineages to exchange genetic material, increasing the chances of new variants emerging.
So it's like those travel hubs are also hotspots for viral evolution, potentially accelerating the emergence of new variants.
That's one interpretation. And the fact that those variants often emerged when the AI was having a hard time classifying genomes by region,
Right.
that increased indistinguishability,
Yeah.
strengthens the link between travel, viral mixing, and the emergence of new variants.
It all seems to fit together, doesn't it?
It does.
More travel, more mixing, more opportunities for viruses to evolve and potentially cause problems.
Yeah.
It paints a rather sobering picture of the challenges we face in a globalized world.
It certainly underscores the need for global cooperation and coordinated efforts in public health and disease surveillance.
Okay, I have to admit, this GMNA method is pretty impressive.
It is quite innovative.
But a part of me is still wondering, how reliable is it? We're basing all these conclusions on the AI getting things wrong.
Yeah.
What if those misclassifications are just flukes?
That's a valid question.
Yet,
And the researchers were very aware of the need to validate their findings and demonstrate the robustness of the method. They took several steps to ensure that these results weren't just random noise.
like what? What did they do to confirm that these patterns were meaningful?
First, they tested the method using different types of AI models.
Oh, okay.
Remember, GMNA is flexible. It can work with various models like naive Bayes classifiers
Right.
or convolutional neural networks.
Yeah.
If the results are consistent across different models, it strengthens the evidence that the findings are real and not just an artifact of a specific AI model's quirks.
So it's like getting a second opinion from a different doctor to confirm a diagnosis.
Exactly.
What else did they do?
They also compared the actual misclassification network to a randomized version of the network called a configuration model.
All right.
In this configuration model, they kept the overall indistinguishability scores for each country the same,
Right.
but shuffled the connections between countries randomly. Imagine taking that global flight network and just randomly redrawing the flight routes.
So it's like they created a what-if scenario where travel patterns were completely random.
Exactly. Now, if the spatial clustering they observed in the real misclassification network was just due to chance,
Right.
you'd expect to see similar clustering patterns in this randomized configuration model as well. But that's not what happened.
Really?
The configuration model didn't show those same strong geographic clusters.
So the fact that those distinct geographic clusters only appeared in the real misclassification network, not the randomized one, adds weight to the idea that those patterns are meaningful and not just random noise.
Precisely, it provides further evidence that those misclassifications are capturing real-world phenomena.
I'm starting to see how powerful this GMNA method can be.
It is a valuable tool.
It's like those AI misclassifications are little breadcrumbs, leading us to a deeper understanding of how viruses are evolving and spreading in our interconnected world.
It's a fascinating way to look at the data.
But what does all this mean for us? How can we use this knowledge to protect ourselves from future outbreaks?
Those are great questions and ones that we'll explore further.
We've been on quite a journey with this deep dive, haven't we?
We have.
We've seen how AI can make mistakes,
Right.
and more importantly, how those mistakes can be like incredibly valuable.
Yeah.
This GMNA method is revealing all sorts of things about how viruses evolve and spread, especially in a world crisscrossed by airplanes.
It is.
But I have to ask, what does it all mean for us? How can we use this knowledge to, stay ahead of the next pandemic?
That's the million-dollar question, isn't it?
It is.
And while this research focused on SARS-CoV-2,
Right.
the implications extend far beyond any single virus. This approach to data analysis,
Yeah.
embracing those AI misclassifications,
Right.
could revolutionize the way we understand all sorts of complex systems,
Wow.
from biological processes to financial markets.
So it's not just about viruses. It's about a whole new way of thinking about data and finding those hidden connections.
Exactly.
I love that it's like we've been so focused on getting the right answer
Right.
that we've been overlooking the richness and complexity of those wrong answers.
That's a good point.
And this opens up a whole new world of possibilities.
It does.
Imagine using GMNA in healthcare, for example.
Okay.
We could analyze misdiagnoses to understand why certain conditions are frequently missed, potentially leading to, earlier detection and better treatment outcomes.
That's a great idea.
Wow, that's incredible application.
Yeah.
It's like we're learning to listen to those whispers in the data, those subtle hints that might point us in unexpected directions.
Well, I like that analogy.
And it makes you wonder, what other areas could benefit from this kind of analysis?
Oh, the possibilities are endless.
Right?
Think about climate modeling, for instance.
Okay.
We could use GMNA to analyze mismatches between climate models and real-world observations to, refine our understanding of climate change and improve our predictions.
Or what about using it to study the evolution of antibiotic resistance in bacteria?
Right.
We could identify new drug targets or
Yeah.
develop strategies to slow the spread of resistance.
Exactly. The key takeaway here is that GMNA is a versatile tool,
Yeah.
a new lens through which to view the world and uncover connections that might have remained hidden otherwise.
It's a powerful reminder that sometimes the most valuable insights come from unexpected sources.
It is.
This deep dive has really challenged my assumptions about what constitutes good data and how we should approach analysis.
I can see that.
It's like we've been so focused on eliminating errors
Right.
that we've forgotten to ask what those errors might be trying to tell us.
It's about embracing those unexpected results and asking those why questions.
Right.
Why is the AI making this mistake? What does this tell us about the system we're studying? It's about fostering a sense of curiosity and being open to new perspectives.
Right. It's about being comfortable with not having all the answers, isn't it?
I think so.
It's about embracing uncertainty and allowing ourselves to be surprised by the data.
I couldn't have said it better myself.
This research is a testament to the power of, interdisciplinary collaboration.
It is.
Bringing together expertise in genomics, artificial intelligence, network science, and epidemiology to address a complex challenge.
Absolutely.
And it highlights the importance of asking those big, ambitious questions, even if the answers might not be immediately apparent.
Right.
Who would have thought that AI mistakes could lead to such profound insights about viral evolution?
It's a testament to the creativity and ingenuity of the researchers involved.
It is.
They took a risk, they explored uncharted territory, and they came up with a truly groundbreaking approach.
Well, fellow deep divers, it seems we've reached the end of our journey for today.
It has been quite a journey.
We've gone from the basics of comparative genomics to the cutting edge of artificial intelligence, all thanks to those insightful AI mistakes.
I enjoyed it.
I hope you've enjoyed this exploration of GMNA and the fascinating connections it has revealed.
Me too.
Remember, the next time you encounter an unexpected result, don't dismiss it as an error.
Right.
Instead, ask yourself, what might this be trying to tell me? What hidden connections might I be missing? Keep exploring, keep questioning, and keep diving deep.